Good afternoon, everyone. Hello, my name is Sadie Junkins. How is everybody doing today? Good. Well, I have an important question to ask everyone. Do you want to do biological research? If the answer is yes, stick around. Me and my friend Jacob Sellers are going to tell you how. So, why should you watch this video? Well, as you can see from this kid's big smile, science is fun. And you take lab classes that show you that. But these lab classes aren't all they seem. It takes 3 hours to do an experiment that you know the answer to. In real research, it takes time and dedication. So usually people pick a gene and find out its function. Let's talk about that. Let's look at designing the experiment. One of the first things you should ask is, what gene do you want to study? And usually that's based on the gene's function. When you pick the gene and its function, you have to look at what model organism you want to use. Here I have a list of model organisms and some of their advantages. But when studying a gene and its function, mice are really close to us, so we usually use them. Now that you've picked your model organism, let's do some knockouts. Not knockouts like punching someone, but knock out a certain part of the genome to be able to stop expression of certain proteins to see how it changes the psychological and physiological aspects of your organism. We delete p38, for example, a stress hormone that is a phosphorylation cascade. And when you delete it and compare to others, you can analyze different diseases. After designing your experiment, you have to perform the experiment. Such an experiment is fasting or stressing your mice to see if that p38 we talked about a second ago is going to be expressed in the same levels or not. We first take mice, grow them up, euthanize or put them down, and collect their liver tissue to analyze these proteins. Once you have your liver tissue, you take mRNA from it. And then from your mRNA, you make cDNA, which is what our bodies do anyway, minus the complementary part. We use a reverse transcriptase polymerase chain reaction, RT-PCR, to do this for us, basically. And it takes a lot of pipetting and time, but very, very cool to do in the end. Once you've got your cDNA and isolated your protein, you can run a gel electrophoresis. You've probably heard of this before, but basically you dye your proteins and you run it with an electric current so they separate out by weight or size. The proteins that you extracted will match a marker protein on the side, and it should be the expected protein that you want in the cytosolic or in the nuclear portion. So watch your weights. So we went over designing and performing your experiment. Now I'm going to hand it off to Jacob Sellers to follow up with collecting your samples. Thank you.